Section 4 of Gray's Anatomy Part 4. This is a LibriVox recording. All LibriVox recordings are in the public domain. For more information or to volunteer, please visit LibriVox.org. Recording by M.L. Cohen. Anatomy of the Human Body Part 4 by Henry Gray. Development of the Nervous System Part 2. The midbrain, or mesencephalon. The midbrain exists for a time as a thin-walled cavity of some size, and is separated from the isthmus rhombencephalide behind and from the forebrain in front by slight constrictions. Its cavity becomes relatively reduced in diameter and forms the cerebral aqueduct of the adult brain. Its basal laminae increase in thickness to form the cerebral peduncles, which are at first of small size, but rapidly enlarge after the fourth month. The neuroblasts of these laminae are grouped in relation to the sides and floor of the cerebral aqueduct, and constitute the nuclei of the oculomotor and trochlear nerves, and of the mesencephalic root of the trigeminal nerve. By a similarly thickening process, its alar laminae are developed into the quadrigeminal lamina. The dorsal part of the wall for a time undergoes expansion, and presents an internal median furrow and a corresponding external ridge. These, however, disappear and the latter is replaced by a groove. Subsequently, two oblique furrows extend medialward and backward, and the thickened lamina is thus subdivided into the superior and inferior colliculi. The forebrain, or prosencephalon. A transverse section of the early forebrain shows the same parts as are displayed in similar sections of the medulla spinalis and medulla oblongata. That is, a pair of thick lateral walls connected by thin floor and roof plates. Moreover, each lateral wall exhibits a division into dorsal or alar and a ventral or basal lamina separated internally by a furrow termed the sulcus of Monro. This sulcus ends anteriorly at the medial end of the optic stalk, and in the adult brain is retained as a slight groove extending backward from the interventricular foramen to the cerebral aqueduct. At a very early period, in some animals before the closure of the cranial part of the neural tube, two lateral diverticula, the optic vesicles, appear, one on either side of the forebrain. For a time, they communicate with the cavity of the forebrain by relatively wide openings. The peripheral parts of the vesicles expand, while the proximal parts are reduced to tubular stalks, the optic stalks. The optic vesicles give rise to the retina and the epithelium on the back of the ciliary body and iris. The optic stalk is invaded by nerve fibers to form the optic nerve. The forebrain then grows forward, and from the alar lamina of this front portion, the cerebral hemispheres originate as diverticula, which rapidly expand to form two large pouches, one on either side. The cavities of these diverticula are rudiments of the lateral ventricles. They communicate with the median part of the forebrain cavity by relatively wide openings, which ultimately form the interventricular foramen. The median portion of the wall of the forebrain vesicle consists of a thin lamina, the lamina terminalis, which stretches from the interventricular foramen to the recess at the base of the optic stalk. The anterior part of the forebrain, including the rudiments of the cerebral hemispheres, is named the telencephalon, and its posterior portion is termed the diencephalon. Both of these contribute to the formation of the third ventricle. The diencephalon. From the alar lamina of the diencephalon, the thalamus, metathalamus, and epithalamus are developed. The thalamus arises as a thickening which involves the anterior two-thirds of the alar lamina. The two thalami are visible for a time on the surface of the brain, but are subsequently hidden by the cerebral hemispheres which grow backward over them. The thalami extend medialward and gradually narrow the cavity between them into a slit-like aperture which forms the greater part of the third ventricle. Their medial surfaces ultimately adhere in part to each other, and their intermediate mass of the ventricle is developed across the area of contact. The metathalamus comprises the geniculate bodies, which originate as slight outward bulgings of the alar lamina. In the adult, the lateral geniculate body appears as an eminence on the lateral part of the posterior end of the thalamus, while the medial is situated on the lateral aspect of the midbrain. The epithalamus includes the pineal body, the posterior commissure, and the trigone habenula. The pineal body arises as an upward evagination of roof plate immediately in front of the midbrain. This evagination becomes solid with the exception of its proximal part, which persists as the recessus pinealis. In lizards, the pineal evagination is elongated into a stalk, and its peripheral extremity is expanded into a vesicle in which a rudimentary lens and retina are formed. The stalk becomes solid and nerve fibers make their appearance in it, so that in these animals the pineal body forms a rudimentary eye. The posterior commissure is formed by the ingrowth of fibers into the depression behind and below the pineal evagination, and the trigonum habenulae is developed in front of the pineal recess. From the basal laminae of the diencephalon, the pars mammillaris hypothalami is developed. This comprises the corpora mammillaria and the posterior part of the tuber cinerum. The corpora mammillaria arise as a single thickening, which comes divided into two by a median furrow during the third month. The roof plate of the diencephalon, in front of the pineal body, remains thin and epithelial in character, and is subsequently invaginated by the choroid plexus of the third ventricle. The telencephalon. This consists of a median portion and two lateral diverticula. The median portion forms the anterior part of the cavity of the third ventricle, and is closed below and in front by the lamina terminalis. The lateral diverticula consist of outward pouchings of the alar laminae. The cavities represent the lateral ventricles, and their walls become thickened to form the nervous matter of the cerebral hemispheres. The roof plate of the telencephalon remains thin and is continuous in front with the lamina terminalis and behind with the roof plate of the diencephalon. In the basal lamina and the foreplate, the pars optica hypothalami is developed. This comprises the anterior part of the tuber cinerum, the infundibulum, and the posterior lobe of the hypophysis and the optic chiasma. The anterior part of the tuber cinerum is developed from the posterior part of the floor of the telencephalon. The infundibulum and posterior lobe of the hypophysis arises as a downward diverticulum from the floor. The most dependent part of the diverticulum becomes solid and forms the posterior lobe of the hypophysis. The anterior lobe of the hypophysis is developed from a diverticulum of the ectodermal lining of the stomadeum. The optic chiasma is formed by the meeting and partial decussation of the optic nerves, which subsequently grow backward as the optic tracts and end in the diencephalon. The cerebral hemispheres arise as diverticula of the alar lamina of the telencephalon. They increase rapidly in size and ultimately overlap the structures developed from the mid and hindbrains. This great expansion of the hemispheres is a characteristic feature of the brains of mammals and attains its maximum development in the brain of man. Elliott Smith divides each cerebral hemisphere into three fundamental parts, that is, the rhinencephalon, the corpus striatum, and the neopallium. The rhinencephalon represents the oldest part of the telencephalon and forms almost the whole of the hemisphere in fishes, amphibians, and reptiles. In man, it is feebly developed in comparison with the rest of the hemisphere and comprises the following parts. That is, the olfactory lobe, consisting of the olfactory tract and bulb and trigonum olfactorium, the anterior perforated substance, the septum pellucidum, the subcallosal, supracallosal, and dentate gyri, the fornix, the hippocampus, and the uncus. The rhinencephalon appears as longitudinal elevation with a corresponding internal furrow on the undersurface of the hemisphere close to the lamina terminalis. It is separated from the lateral surface of the hemisphere by a furrow, the external rhinal fissure, and is continuous behind with that part of the hemisphere which will ultimately form the anterior end of the temporal lobe. The elevation becomes divided by a groove into an anterior and a posterior part. The anterior grows forward as a hollow stalk, the lumen of which is continuous with the anterior part of the ventricular cavity. During the third month, the stalk becomes solid and forms the rudiment of the olfactory bulb and tract. A strand of gelatinous tissue in the interior of the bulb indicates the position of the original cavity. From the posterior part, the anterior perforated substance in the piriform lobe